Welcome to the GMO Freakshow. Here's your ticket. Enjoy the show.
Give me your ticket, please. Right this way. Welcome to our freak show.
Come in, please. Look, I have traveled the 7 seas just for you. See here, my perilous journey begins. And of course, for the fabulous treasure. Here we start and we begin in 1984. Of course, that was the year where there was big hair, beautiful clothes, and also fantastic music. Now, in this time period, there happened to be many scientists casually talking about. They decided, well, of course they were talking about, well, science, because they have nothing better to do with their lives. So, it all began with a man named Peter Jennings. And he proposed a question to many of the scientists that were gathered round, and he said, do you know what my favorite GMO is? And of course they looked at them the exact same way you're looking at me right now. And he said, I don't care what you think. My favorite is the yellow, what is it? Yellow endosperm. Yes, that's his favorite GMO. And do you know what makes yellow endosperm yellow? Well, of course, it's beta-carotene. It's the same thing that you find inside of corn that makes corn yellow. So, beta-carotene contains a beautiful vitamin called vitamin A. And he injected this into the rice. Of course, this is the Taylor golden rice. He injected it in there. And he decided that because vitamin A is so valuable and used for vision, that it would of course cure blindness and of all sorts. Now, 2 famous scientists from Switzerland, however, don't ask me to pronounce their names because I cannot do it, but they decided to, collaborate a little bit on Jennings' discovery for the IR8 rice. They decided, well, if you're going to make something, you might as well make it useful. And so they added iron. Now, iron, as some of you may know, is something that is in the blood and is needed in order for oxygen intake to process in the immune system. Now, with that being said, they decided, when they collaborated with them, that they would have to help somebody. Somebody could possibly find this useful, and they thought people in poverty. People in poverty could find the rice useful because some of those people in poverty, such as the ones in Asia and the Philippines and Africa, could probably use it because that is their main source of, in their diet, is rice. So, with that being said, because of their efforts, their goodwill efforts, they gained much popularity. One, in this era, 2015, they were presented with the Patriots for Humanity Award, which is very close to the Nobel Peace Prize. Not to mention, Pope Francis himself graced all of them with their presence and blessed the golden rice, in hopes that it would gather on and it would continue and become successful. Also, the Helen Keller International has also took it into consideration and has put forth its efforts because of their efforts to help with blindness and vision. However, as you may notice from my map, not all journeys are without their perils. And this one, my friends, sure has been. For one, many scientists have faced a very ethical issue, one of which happens to be when they first were testing the product. Now, they made one minor mistake when they were trying to, introduce it. They decided to go to China and introduce it to Chinese children. Their mistake was they had forgotten to let the Chinese government know that they were doing this. Of course, it was maybe a minor mistake. You be the judge of that. But that, of course, did not sit well with the Chinese government, and it was caught wind by the Philippines, which they were already starting to grow the crop there and establish a little bit of prosperity with this crop, with the golden rice. However, soon people, because of what happened in China, they decided that GMO rice was bad. This golden rice, not good. So they'd vandalized the whole field. They just slaughtered it and they just pulled it up from their roots. And because of that, you know that was caused by fear. Fear of things they did not understand. And that, my friends, is why many scientists are being ridiculed for putting GMOs in food. Now, as you know, many stores have, grown into no GMO writing on their boxes and whatnot. And also, a lot of people, naturalists, believe that it is not a good thing. And it could be lack of education, but one of the reasons is also because many people fear that it has health risks. In other words, like, allergies could be spawned from GMO and uprise from, within the depths of themselves. And not to mention, they also maybe affect medications a little bit, wavery. And also, they possibly... The biggest reason is because scientists have not studied it enough yet. And because they have not studied it enough yet, it leads to the question of, do they even know what they're doing? Yes. And it's a very scary thought. Now, another big fear that a lot of people have had is that they cannot contain the whole essence of the crop because it's sporadic. I mean, it's a crop. Animals will swoop in and they will gather it and they'll carry it away, or the wind might blow it off and it'll go into some other environment and affect the biomes there. That's why they are afraid, because they don't know how it'll affect the environment. Also, another thing is that it's a minor thing, but of course, few people with their selfies nowadays may find it a little concerning. It turns skin, too much beta carotene turns skin yellow. Oh, I'm not talking about the jaundice yellow. And not looking like a Simpsons character either. I'm talking just a tad bit of yellow. Just look. But, everybody's, so egocentric nowadays. It may be a big deal to them. So, anyway, as we've looked at all these perils that I have journeyed to bring this to you, I now present to you what lies at the end of the X marks the spot, as we know is what all pirates look for. And that would be the golden rice. Yes. One of the greatest treasures of the world, especially what it is able to do for people in need, especially those in poverty in Asia, in the Philippines, as well as in Africa, if we can span out that far. Now, the biggest thing that scientists want to do, their main goal, their X, is to produce a crop yield that allows the prosperity of the poor economy, so that it, gathers more and they can fend for themselves and they can be able to produce more of this. As well, they decrease in malnutrition, anemia because of the blood and the iron in the rice, and also to help cure blindness, if possible. Now, another big thing, and that is the purpose of this whole entire freak show, is it makes people aware of exactly what GMOs are and their benefits, that not all of them are exactly as they seem. Now I know I said that there wasn't very many studies being done. However, there is one that has made a staple in their whole process of what they've done. It is that one simple bowl of this rice can supply 60% of the nutrients children need. Yes, you heard me right, 60%. Now with that in mind, think about how much that could benefit the prosperity of the children in poverty. It would allow them to be able to be prosperous later on and for future generations to come, since by age 5, all children are developing a very stable immune system and growth and things that they don't understand what's going on right now. But if many scientists can fulfill their dream of reaching that X, so can this be for the tale of the golden race. Thank you.
Talking about BT corn and whether or not if it's worth the risk. So first off, let's talk about what a GMO is. A GMO is a plant or an animal that has been modified with a small amount of another organism, and it's through molecular techniques. So GMOs have been given this genetic trait to hoard off pests and tolerate pesticides as well as give quality. So the BT corn is a genetically modified organism, and the transgenic crop has a pathogen of insect pathogens in it that is called Bacillus thuringiensis, and it encodes protein that kills off the European border, which is a pest that eats and destroys corn. So, how to create BT corn? The gene produced from... I just lost my train of thought. Okay. To create BT corn, the gene produced from the genetic trait of interest gets separated from the donor organism, which the donor organism is the naturally occurring soil of the BT corn. So the protein that is produced to kill the insects such as moths, butterflies, beetles, bees, and so forth, it is insected into the corn, and once the insects eat it, they will soon die off. So when the BT is insected into the gene, into the corn, the European borders will eat it, and within 2 hours of their first bite, they will stop feeding. Now, once they stop feeding, the crystal-like protein that is produced into the BT corn is a poison that is into their stomach, and it will actually kill them within 2 to 3 days. Now, the European borders have 4 stages of life. First, they start off with their full-grown larvae, and they hibernate in corn stalks. And this is through the month of November and April. Now, once it hits spring and the weather exceeds to 50 degrees, they will start the spring production again. So they start off with their full-grown larvae and transfer to their pupa into their moth form, and then they restart from their eggs to the larva to the pupa to the moth form. This consists of 1 generation. Now, in the early European corn borers, the first generation was normally discovered for 1 year. Now, in the late 1930s, the second generation of European corn borers started to be seen, and they were actually growing very rapidly and became very dominant. Now, in the third and fourth generations, they were spreading through the north central area into the eastern part as well, just pretty much like lining the coast of the United States. And now today, the European corn borers actually populate for the 7 most western continental states. BP corn is actually found in multiple things that we eat within our natural occurring day, such as popcorn, high fructose corn syrup, and soda, baked goods, your tortilla chips, your pizza crust. Just about everything has BP corn in it. And 90 to 95% of all United States corn crops and even soybean crops are GMOs. So GMOs are everywhere, and let's see. We're not the first guinea pigs to actually try out the BT corn. It's FDA approved, it's safe for humans, fish, birds, mammals, and even the environment through selectivity because it's actually an insect pathogen, so it's not harmful to humans. It's been around since the 1960s, although there are a lot of allergies that have been recorded from BT corn, but BT corn is actually an alternative to spraying with pesticides. So the Roundup Ready corn that's actually around here in Warsaw is a lot worse than the BT corn, and BT corn is one of the best alternatives for the GMO corns.
Welcome to the glowing cat exhibit. You're probably wondering, why a glowing cat? Well, besides the reason for science, researchers from the Mayo Clinic were looking for a way to, the best way to genetically insert certain genes in animals. There are certain primates that are immune to the AIDS virus, since their immune system cells have antiviral proteins. They prevent the virus from replicating. The main purpose of this experiment was to determine the best way to transfer these genes into an AIDS-susceptible species. Cats were chosen as the species. They are one of the few that are susceptible to AIDS. Cats and humans share a similar genome, and they're easy to acquire, take care of. There's not too much issue in raising cats. To determine that the genetic insertion was successful, they also wanted to transfer the gene that codes for the green fluorescent protein. The logic being, if the cat glows in the dark, the genetic modification was a success. There were different methods of genetic insertion that were considered. Cloning was the first consideration. However, cloned animals tend to have many genetic abnormalities, and they don't tend to live a healthy or very long life. Injection of modified embryonic stem cells into the blastocyst was also considered, but acquiring adequate embryonic stem cells for this particular experiment was problematic. So they decided to go with microinjection of a viral vector into an oocyte. A viral vector is a virus whose genes that allow it to replicate and cause harm are removed, and the genes that they were going for, in this particular case the green fluorescent protein and antiviral proteins, were used to substitute them. The viral vectors were infected into cat oocytes. The oocytes were fertilized via the in vitro method, and then the embryos were inserted into the exposed fallopian tubes of female cats in heat. To stimulate ovulation and get the process going, they used a vasectomized male cat. The result of 22 embryonic transfer procedures was 5 pregnancies and 3 live kittens. All 3 kittens were successfully transgenic. All 3 were fluorescent, and it was determined that their immune system cells had the antiviral proteins at the end of it. Now, they wanted to see if the antiviral proteins allowed the immune system cells to fight off FIV, or the feline immunodeficiency virus. So they put them both in a petri dish of sorts and recorded the results. Unfortunately, the immune system cells were only partially immune to the FIV. They weren't able to completely eradicate it. However, it's a step in the right direction. The researchers also wanted to see if these genes can be passed on to offspring. So once one of the male cats was fully grown, they bred him with 3 normal females. All of its progeny were also transgenic. The genes were successfully transferred over. Now, with genetic modification, we run into some ethical issues. Most researchers across the globe who do research on animals agree to follow the 3 R's: replace, reduce, and refine. Replace refers to the search for alternatives to animal testing. If you can find a way to do your experiment without the use of animals, please do so. If you have to use animals, find ways to reduce the amount of animals that are being used in the research, and refine refers to the change in techniques to minimize animal suffering and prolong human life in the cats, or well, in this case, the cats. In this particular experiment, there were also some theoretical ethical concerns. One of them being, did we have the right to change the genome of the cats? Were the cats' rights infringed on? Did they have any rights to begin with? There were some unintended consequences of the experiment, and then finally there's the justification for the experiment itself. One of the cats, which was a male, was afflicted with some health problems and genetic abnormalities. During his upbringing, he suffered from a lower eyelid irritation and an abdominal hernia. He was also suffered from cryptorchidism unilaterally, which just means one of his testicles didn't drop. He also had sporadic pruritic dermatitis. Every so often, his skin would get red, itchy, inflamed, similar to an allergic reaction. It was unclear if these complications were due to the genetic modifications, but they don't match a known syndrome. 2 cats also died during the birthing process, with one dying from aspiration complications. And one of the pregnancies also ended in 5 miscarriages. The fetuses died 10 days before they came to term. Dissection of the deceased resulted in no defects being found, and like before, it's unclear if the genetic modifications caused the problems to begin with. The researchers in question did follow the 3 R's to the best of their abilities. Based on the types of experiments that they were doing, it was determined that animals were needed for this particular experiment. Any alternatives would not have been viable. To reduce the amount of animals that were used, they acquired the sperm and egg cells from the gonads of neutered and spayed cats. And as for the kittens themselves, they were fed, they were taken well care of, socialized, played with as they were grown up. They were not stuffed in a corner and forgotten about. The research could give us a better understanding of the HIV disease model, and gene therapy treatment could eventually be created with the use of restrictive and antiviral proteins. Unfortunately, since 2011, there has been no progress in this line of research or any updates. However, there have been continued research in the study of HIV using cats. Back in 2013, Dr. Jeanette Yamamoto and her fellow researchers at the University of Florida found that T cells from HIV-positive individuals were able to bind with a certain peptide region of the FIV and deactivate it. The problem with any vaccine in the past with HIV is HIV has a tendency to mutate. However, they were able to bind to the exact same peptide chain each time. This could mean that this peptide chain, despite mutation, would remain the same consistently, as in it's a chain that cannot mutate for it to be FIV or HIV. With continued research, they hope to produce an HIV vaccine in the next few years.
Welcome to the wonderful world of plants. Can plants map out explosives? That is a good question. That is the same question that Neil Bruce and his team of researchers tried to figure out after doing some research with several mustard plants until they found one species that was lacking the MDHAR6 enzyme, which normally recycles vitamin C. They took this plant with this missing gene and spliced it together with a bacteria that could essentially help clean up explosive residue from the soil. What this plant does is once it's planted, it absorbs the residue through the roots, getting rid of the pollution matter and helps clean up the soil. Then the plant can be removed from the soil and disposed of to get rid of the residue. Why is this process important? It can be done with several other plants like switchgrass, algae, tobacco plants, and other mustard plants. With the tobacco and algae plants, the research has been done in China where they took a rat gene that produces metallothin, which attracts metal residue from mercury, and then the plant is able to absorb it through the roots and it gets trapped within the cell wall. So then they can dispose of the plant in a laboratory in a safe manner so it doesn't go back into the environment. This is a helpful process because it can help get rid of pollutants in the soil that normally ruin good microbes for crops, and it also helps save the lives of animals that wouldn't intake in the mercury found in water. We also have a featured company by the name of Ulex, which is doing some genetic modification of their own. They have taken the aster shrub that is normally found in the southwest region and modified it to find several different species in order to find one that can yield the most rubber. The one that they have found that can do this the best is from the wily plant, which is a shrub that they can cut from the bottom midsection and leave the root within the ground for it to grow again, which the process takes about 1 to 2 years for it to maturely grow again. With the portion that they cut out, they can put in a solution which gets the rubber out, and then they can produce tires, latex gloves, and balloons. This form of rubber is different than the one we normally find in rubber trees because it doesn't contain 2 proteins that normally cause allergic reactions from latex, which is a huge benefit for people who have allergies to latex products. And also, the chemical bond within this type of rubber is a lot stronger than the one you would find in the natural rubber tree. And this will be very helpful because then we can grow our own rubber plants here in the United States rather than having to import from overseas to produce our own tires and other rubber materials. This company, Ulix, has also partnered up with Cooper Tire and the University of Arizona to further the research so that way they can mass produce this later on. And this, again, can be very beneficial just because we can do several products out of this rubber. And it's also been tested with wetsuits that are lighter and more flexible for surfers.
Come in, and I will tell you all about my pet here. My pet, the spider goats. The spider goats that I found research on in a top secret file. In the year 2000, a company named Nexia Biotechnology tried to find a way to mass produce silk. The problem is, silk is produced by a certain spider called the orb-web weaver spider. This spider is a cannibal. So, for them to mass produce this silk, they would have to have a spider farm, which would pretty much result in the spiders simply eating each other and not producing any silk. Instead, they decided to look for another animal that could possibly produce the silk in replace of the spider. They found this animal in the goat. The protein that the goat uses to produce milk is similar to the protein that a spider uses to produce silk. And so they took the DNA from the spider and inserted it into the embryo of a goat, and then put that embryo inside of a mother goat. From there, they had 7 goat children born. One of those goat kids was named Freckles. And when Freckles became a mature goat, they milked her. They skimmed the fat off of the Freckles' milk, raked through it, and pulled up silk strands out of the milk, which were then dried and processed. Out of the 7 goats, 3 became silk-producing goats. A few years after this discovery, Nexia Biotechnology went bankrupt and was out of business, but their herd was bought by a professor of genetics named Randy Lewis from the University of Utah. There, they have a farm where they keep animals for animal husbandry classes and genetics classes. Now, today, from the 7 goats that they purchased, they have a herd of 50 spider goats. Of these 50 spider goats, 2/3 actually produce silk through their milk. The other third is left for reproductive purposes because the spider goats have now inserted the spider DNA into their own DNA, and the spider goats are born regularly. Because of the extra protein in the goat milk, this type of silk is actually stronger and has more elasticity than the spider silk. This is very beneficial because it can be used in several different types of materials. For instance, they are testing them to make ligament and tendon replacements eye sutures, jaw repair. It is being tested in military purposes for Kevlar jackets, bulletproof vests, and even parachute ropes and climbing gear. Domestically, it's being used to test on airbags for vehicles, harnesses, and nets. Because of this, there's been a lot of research on what the effects on the goats are. So far, there have been no physical or behavioral differences in the goats. There have been no health issues that have risen from this situation either. For the future of this experiment, they've actually been looking for other ways to produce even more silk, and they have found a way with alfalfa plants. Alfalfa plants also produce the same type of protein, and so they will be trying to insert spider DNA into the alfalfa plants to create a silk farm. The impact of being able to produce so much silk without having to scurry around searching for spiders can impact the cost of medical care, because the more abundance of silk we have, the less the sutures and the tendons will cost. And who knows, someday we may even end up with the miracle of a spider goat man. Thank you.
Okay, ready? Go for it.
Yes, come further.
Sorry. That was me. I kicked it, so it went like that.
I thought you got scared. I'm like, I'm sorry. All right, I'll stop. Oh my God. You were scared. I was